Good afternoon. My name is Regina Vontell. Um, Thank you for coming, and I would like to thank the committee for putting on such a wonderful conference. I have one disclosure, and that is many of the authors, the work I'm presenting, are on the patent for ASC molecule IC100 antibody. So today I'm going to talk about neurogranin and inflammatory mediation through the inflammasome complex. And what we've done here is correlated this with the hyperphosphorylated tau in postmortem brains samples with Alzheimer's disease. So this is low Alzheimer's disease and intermediate Alzheimer's disease changes. So we compared the two, and we're looking at how does tau change and how do these other proteins change as tau hyperphosphorylates. So Alzheimer's disease has some particular pathological features. It's a progressive neurodegenerative disorder, and it has clinical, biological, and pathological features seen in postmortem. And this goes along a continuum. So it's not only done through the hippocampus. We know that in order to do a BRAC scoring, you have to have about 20 different samples, and you look to the extension of the pathology of both beta amyloid and tau. But the group that we're mainly interested in is the low AD, which will show hyperphosphorylated tau around the central cortex and subiculum and CA1. Now, as this changes from low to intermediate, you start to see tau proteins in CA2, 3, and the dentate gyrus. But the volume of the hippocampus itself was not significantly different, which gives us an opportunity for possible interventions. And intracellular, we're looking at the neuron and the changes that it shows post synaptically, and these synapses and the plasticity. But also we're looking at what happens with the microglia as they're surveying and finding these inflammatory mediations. They're going to go from surveying to amoeboidal. So when we want to consider plasticity, we consider neurogranin. It's a protein kinase C substrate that binds with calmodulin in the presence of calcium. So this is going to be in a hyperphosphorylated form. It's triggered by the binding of glutamate NMDA receptors, and it's a postsynaptic protein involved with long-term potentiation. And so it's really thought to be a cascade for postsynaptic transmission and synaptic plasticity. So clinical studies, we did one study, clinical studies show that the concentration of CSF and plasma neurogranin increases as AD progresses. And so that we know that this is important for long-term memory, and we know that this is a great biomarker for Alzheimer's disease changes. So we're also interested in inflammatory signals. So we want to know what happens between the neuron and the microglia, and what's going on inside the neuron and what's going on in the environment itself. So one inflammatory mediator that we're very much interested in is this inflammatory complex. And this is relatively a simple molecule that's designated in this windmill formation. And it's activated by a nucleotide binding oligomerization domain protein, or a NOD-like receptor. A NOD-like receptor, and we've seen it in 1 and NOD-like receptor 3, triggers the oligomerization of ASC, which is apoptosis-associated speck-like protein containing a caspase recruitment domain, or a CARD scaffolding protein. This goes on to cleave caspase 1, procaspase, into its active form, and that in turn turns the activation of the proforms of cytokines IL-1 and IL-18 into its active form. But it also activates the pyridine domain. Well, this is important because we've seen in other studies that in neurons, they seem to be activated by NOD-like receptor 1, whereas in microglia, they're activated by NOD-like receptor 3. I'm sorry, I turned this too quickly. And so we've also looked at the different domains of ASC, and we recently published that in 2023, where we showed that if it's activated, if one looks at an antibody raised in the card domain, you see it mainly in microglia. Whereas if you look at an antibody raised in the pyridine domain, we see this in neurons. So this is where the IC100 is very interested. For this study, we wanted to analyze the neurogranin MAP protein tau, beta amyloid, and the ASC, both the monoclonal, which is raised against the card domain, and the IC100, which is again raised against the pyridine domain. So we wanted to see what are these proteins doing in intermediate and age-matched controls. And then we wanted to look at what is the difference of just neurogranin itself in these two cohorts. And we examined the relationship between neurogranin and tau and beta amyloid. And then we wanted to do some quantitative studies of looking at the neurogranin in the two different ASC molecules. So this paper was very interesting, and we found some really interesting results with this study. So in order to do this, we used the same cohort as we did in the 2023 paper, where we looked at the different ASC activating molecules and the inflammasome complex. We used 17 age-matched controls, meaning that there was low AD neuropathological changes. And again, low AD changes mean that the beta amyloid, you can be any amounts, they didn't really significantly change. But the p-tau, the phosphorylated tau, again, you can see it here in the entorhinal cortex and a bit in the CA1. This change from low to intermediate, where again you can see amyloid clusters, but right up, even grossly, you can see that there's a change in the tau level. So we looked at both their clinical review before they died, and normally we would have either a TICS or a MoCA score. And then we reviewed the neuropathology report to put these two groups into different cohorts. And this is available on the Vontell et al. 2024 paper. So what did we find? We found some interesting things. Obviously, morphologically, they look a bit different. In our what we call low AD or control group, you see that there's some neurofibrillary threads. The MAP2 showed that you can see the apical dendrite in these large neurons. And then when we looked at neurogranin in the controls, you can see that this pretty much follows suit into what MAP2 shows. When you look at intermediate AD, of course you have more neurofibrillary tangles, so you start to lose MAP2 staining, and also there seems to be a loss of neurogranin, and that may be very important because it shows how this is a biomarker. And why is it going into the CSF? Why do we see it in the blood plasma? Well, it may be because it's spilling out of these neurons. And so we did a correlation between neurogranin and the increasing of tau. So as we saw, as tau increases, you have a loss of neurogranin, and they showed a significantly different negative correlation in the entorhinal cortex, CA1, CA2, and CA3 regions, and also the dentate gyrus. So the next thing that we wanted to do was to see how neurogranin negatively correlates with the ASC expression in microglia. So again, this is going to be the inflammasome main component, the oligomerization of ASC, but it's going to be against the card domain. And what we found is that when ASC, if using an antibody of ASC that's raised against the card domain, you can see it mainly in this microglia. And IC100 is the antibody raised against the pyridine domain. So these are neurons that are going to have some tau, they're going to have some neurofibrillary tangles, and you can see that the yellow represents the ASC 100 is in red, but you can actually see that the microglia are surrounding them. And when we looked at the loss of neurogranin and the correlation of ASC, again, we saw that as you lose neurogranin, it correlates with the increasing ASC expression. So we were very excited about this, which means that as the neurons also phosphorylating and changing morphology, it's actually having an immunological response. So when we look at the ASC antibody, the humanized ASC, and we look at the neurons, because we found it mainly in neurons, we also see a negative correlation. As you lose neurogranin, as you gain ASC, or as neurogranin loss, there's an increase in ASC. And this negative correlation could be seen in CA1 and CA2, but not necessarily in CA3, which means that this intermediate step, which will go along with mild cognitive loss to some intermediate cognitive loss, shows that there's actually an open window to use this antibody. So it may be a therapeutic target to help alleviate some of the conditions. So in conclusion, we have identified the morphological expression of MAP2. We wanted to use not only neurogranin, but another MAP2 molecule to look at these changes. And we have identified that these neurons change in the presence of intermediate AD and compared to their control cohort. So expressions of the postsynaptic neurogranin can be seen in different subfields of the hippocampus. So we saw that there seems to be more neurogranin loss in CA2 and 3, but CA1 in both groups, both the intermediate and AD, are very similar in morphology. So we weren't too surprised about that. Neurogranin expression was altered in AD cases compared to the controls. It's expected to lose neurogranin, and this is one of the ways that we showed that this could actually be a wonderful biomarker for dementia, and it's now being considered for that. And then we also saw a negative correlation of neurogranin across all the three hippocampal subfields using the anti-mouse ASC. Again, this is going to be erased against the card domain. So this shows that during the progression of AD pathology, tau aggregates form, right? We know this, and a beta amyloid also forms in the diffuse diffuse neuritic plaques. And so we think that as this parenchyma changes, it's going to make the microglia toxic, and it's going to aggravate them, and they're going to be more reactive. We have a lot more work to show that, but it's the beginning of this. So as AD progresses from CA1 to the other subfields, we're seeing that this ASC is going to oligomerize in microglia. This is more inflammatory mediation that probably needs to be shut down in order to prevent the continuation or the progression of dementia. And then we also interestingly found that as neurogranin loss, it's in concert with the increase of neuronal ASC. So the IC100 increases as you lose that neuronal plasticity. So this means that the neurons are a bit more unstable, probably because of the phosphorylated tau. But also that means that in the neurons, they're having inflammatory processes, and they're actually recruiting ASC molecules. And so this may show that there's a temporal progression of AD. Again, we looked at the hippocampus, which is a wonderful place to start with Alzheimer's disease, but the work now is to look at other regions of the brain, look at other regions that we don't expect to be affected by intermediate AD, such as the peristriate area or the occipital, and to look to see how much of this protein and how much of this inflammatory complex is oligomerizing in those neurons, and then do you also lose neurogranin? So it's going to show that there's a synaptic plasticity loss, or does it also increase with the ASC molecules, or which comes first? So we're very excited to continue this work, and we want to also include RNA studies too. So with that, I'd like to thank some of the collaborating labs we had from the University of Gothenburg. There was some wonderful collaborators that provided the Neurogranin antibody, the Miami Project to Cure Paralysis. We have some really wonderful collaborators, Dalton Dietrich, Dr. Keene, Dr. Vicari, and Dr. Bramlett has provided me with the ASC molecule, the IC100, and we're hoping to use that very soon in clinical trials. And then in my own group, I'm from the Brain Endowment Bank of the University of Miami. I'd like to thank my collaborators, Dr. Davis and Dr. Sun, and also a few folks in the brain bank themselves that gave me some important clinical information, helped me with the immunostaining, and helped us with this project and made these 2 papers successful. Well, thank you.